Welcome back to the deep dive. We're here again, sifting through stacks of, well, pretty specialized research to pull out the core knowledge you can actually use.
Exactly. Keeping it clear, keeping it focused.
And today we're doing something a bit different. We're zooming way in, not a whole system, not a major disease category.
Nope.
We are dedicating this entire deep dive to just one term from pathology. It sounds small, maybe, but it's everywhere, and often misunderstood. Hyaline change.
It really does sound obscure, doesn't it?
Oh.
Just one word for how something looks under the microscope.
Yeah.
But getting this one right is actually fundamental.
Fundamental.
Because it's this visual clue, right? This homogeneous, glassy, pink look. And seeing that could mean several very different things are happening biologically.
Okay.
So our mission today, using our sources, is to give you the context behind that look. What's happening at the molecular level? What does it mean clinically? It helps you figure out, okay, is this just a temporary cellular traffic jam, or is it, a sign of serious long-term damage?
Right, so it's about connecting the visual to the underlying process. And you said there's a key aha moment here.
There absolutely is, and it's this. Hyaline is purely descriptive. It describes an appearance. It is not a specific disease itself.
Okay, descriptive, not diagnostic on its own.
Exactly. Think of it like finding, a piece of clear plastic at a scene.
Okay.
Is it from a broken window, a water bottle, industrial waste? You don't know just by seeing clear plastic. You need context, location, what it's made of.
Gotcha. So hyaline is the clear plastic of pathology. We need to figure out what kind of hyaline it is and why it's there.
That's precisely what we're diving into. The context is everything.
All right, let's start right there with the visual, because pathology often starts with what you see down the microscope. When someone says hyaline, what's the image we should have in our minds?
Okay, first, remember the background. Most tissue slides are stained with hematoxylin and eosin, right? H&E.
Right, the classic purple and pink stain. Hematoxylin stains nuclei blue or purple.
Those are the acidic bits called basophilic. And eosin stains most other things, especially proteins in the cytoplasm and outside the cells, pink or red. That's eosinophilic. Hyaline is like the ultimate eosinophilic finding. Our sources define it pretty clearly. It's an alteration either inside cells or in the space between them that results in a homogeneous, glassy, pink appearance with that H&E stain.
Homogeneous, glassy pink, so smooth, uniform, intensely pink. Like if you're looking at a sample, maybe you see a patch that looks like smooth, pink glass instead of the usual textured cells or fibers.
That's a great way to visualize it. Too smooth, too uniform, almost translucent compared to the surrounding tissue. It implies there's a dense buildup of something that's soaking up that pink eosin stain really evenly.
Okay, dense pink glassy. But here's where you said the nuance comes in.
Exactly. We have to stress this. Hyaline is a descriptive histologic term, full stop. It is not in itself a specific marker for one particular disease or even one specific type of cell injury.
Right. I think I'm getting it, but let me push on that a bit. If it looks so distinct, glassy, pink, why doesn't that automatically tell you what's wrong? Doesn't the body only make certain things look like that?
That's a common assumption, but it's the key misconception we need to clear up. The reason it's descriptive, not diagnostic straight away, is because lots of different molecular changes can end up looking like this.
Ah, okay.
It could be a buildup of highly concentrated normal proteins. It could be altered connective tissue. It could be tangled up bits of the cell's internal skeleton. It could even be plasma proteins that have leaked out of blood vessels. They all can end up looking dense, packed, and pink.
So seeing hyaline is like an alarm bell going off. It means look closer, figure out what this pink stuff is and where it is.
Precisely. It forces you to ask, what kind of accumulation am I looking at here? And the location is the first big clue.
Which is how we're organizing this deep dive, right? Based on location.
Yes. We're splitting it into two main categories. Intracellular, hyaline, the stuff accumulating inside the cell, and extracellular hyaline, the stuff accumulating outside the cells.
And that location immediately gives you hints about what's going on clinically.
Absolutely. Intracellular hyaline often tells a story about, say, temporary overload in the cell, or maybe a problem with making or moving proteins. Extracellular hyaline, on the other hand, that often points to more permanent changes, like chronic damage, scarring, or long-term problems with blood vessels.
Okay, let's get into the inside story then. Accumulations within the cell. You mentioned 3 classic examples, often tied to how proteins are being handled.
That's right. And the first one is probably the least severe and most reversible of the bunch, reabsorption droplets in the kidney.
Reabsorption droplets. Okay, the name kind of gives it away. It sounds like the kidney cell is just absorbing too much stuff.
Essentially, yes. You see this typically in kidney diseases where there's a lot of protein leaking from the blood into the urine, what we call proteinuria.
Right, the filter in the kidney, the glomerulus, is damaged and lets protein through when it shouldn't.
Exactly. Now, the proximal renal tubules, these cells lining the kidney's tubes, their job includes reabsorbing any small amounts of protein that do sneak through the filter normally. They do this by drinking the fluid around them, a process called panocytosis. They pull the protein, mostly albumin, inside in little sacs called vesicles.
And normally, what happens to that protein inside the cell?
Normally, those vesicles quickly merge with lysosomes, the cell's recycling centers. The lysosomes break down the protein into amino acids, which the body can reuse. It's a very efficient process, usually.
But if the filter is badly damaged, like in some kidney diseases,
Then, instead of a trickle of protein, the tubule cells face a flood, heavy proteinuria. They ramp up panocytosis, trying desperately to reclaim all that valuable protein.
okay, so they're pulling protein in like crazy.
But the lysosomes just can't keep up. The rate of protein coming in overwhelms the rate they can break it down.
So you get a backlog, a traffic jam inside the cell.
Precisely. These pink hyaline droplets you see inside the cytoplasm of the tubule cell are essentially those vesicles packed with protein, waiting to be processed by the lysosomes, or maybe already fused, but the lysosomes are just stuffed full.
Okay, but wait. If it's just normal protein that the cell intends to process, just temporarily stored because there's too much, why is that considered pathology? Isn't the cell just doing its job under stress?
That's a really insightful question. It is pathology because the underlying cause, the heavy proteinuria, is due to a disease process, like glomerular damage. But you're right to question the severity of the accumulation itself. And this is the crucial point about these droplets. This process is typically reversible.
Okay.
If the underlying problem, the proteinuria, gets better, maybe the kidney disease is treated successfully, then the protein load decreases. The tubular cells catch up, metabolize those stored droplets, and the hyaline appearance vanishes.
So it shows the cell is actually adapting, even if it looks abnormal temporarily.
Exactly. It's a sign of cellular stress and overload, but also cellular adaptability.
Okay, that makes sense. Reversible protein backlog due to intake overload. Now, how does that compare to the second example, Russell bodies? You said these are also protein, but it's a different kind of overload.
Yes, a completely different scenario. Russell bodies are the classic example of accumulating excess, normal secreted protein. They occur in plasma cells.
Plasma cells.
Yeah.
Those are the immune cells that pump out antibodies, right? Immunoglobulin.
The antibody factors, exactly. They are designed to synthesize and secrete huge amounts of immunoglobulin protein. Now imagine a situation where these plasma cells are highly activated, maybe in chronic inflammation or sometimes in plasma cell cancers like multiple myeloma. They go into overdrive.
They're making too much antibody protein.
Way too much. Now, antibodies are complex proteins. They get synthesized and folded inside a cellular structure called the endoplasmic reticulum, or ER. Then they move to another structure, the Golgi, for packaging before being shipped out of the cell.
So what happens when the production line is running too fast?
It jams up. Specifically, the proteins start accumulating within the endoplasmic reticulum itself. The ER network gets massively swollen and distended with all this excess normal immunoglobulin that hasn't been secreted yet.
And that's what we see as Russell bodies.
Exactly. These hugely distended ER cisternae packed with protein appear as these large, homogeneous, eosinophilic inclusions, the Russell bodies. They're basically big, intracellular blobs of stored, unsecreted antibodies.
Okay, so let's contrast. Reabsorption droplets, protein coming in too fast from outside, Russell bodies,
Right.
normal protein being made inside too fast to get out.
You've got it. Two distinct mechanisms leading to intracellular hyaline protein accumulation.
Now, here's a tricky question maybe. How does a pathologist looking at this distinguish Russell bodies, which you said are excess normal protein, from, say, the buildup of abnormal or misfolded proteins that might signal something more sinister, like a cancer? Do they look different?
That's a fantastic question, and it highlights the need for context. Morphologically, just looking at the pink blob, they can look very similar glassy eosinophilic inclusions. Right.
However, Russell bodies are typically composed of structurally normal immunoglobulins, just too much of them. In malignancies, like certain lymphomas or myeloma, you often look for other clues. Are the plasma cells themselves looking abnormal, atypical?
Well,
Is the protein they're making monoclonal, meaning it's all derived from one cancerous clone? And sometimes in cancers, the accumulation might be of genuinely misfolded or mutant proteins that were never going to be secreted properly anyway.
So the presence of Russell bodies in, say, a plasma cell tumor is more a sign of high metabolic activity by those cells,
Right.
but the actual diagnosis of cancer relies on other features, the cell appearance, the clonality, the specific markers.
Precisely. The hyaline inclusion itself is a piece of the puzzle, indicating high protein synthesis, but it's not the whole story for diagnosis. You need the broader context.
Okay, that clarifies things. Let's pivot to the third intracellular example, alcoholic hyaline. This one sounds different already.
It is fundamentally different. This throws a bit of a curveball because we're moving away from proteins being absorbed or secreted.
Okay. Alcoholic hyaline, which is also often called a Mallory body or Mallory-Denk body, is really characteristic of significant liver injury, especially in alcoholic liver disease. It still shows up as a dense, eosinophilic, sort of irregular clump within the cytoplasm of the liver cells, the hepatocytes.
Still pink and glassy-ish then.
Yes, still eosinophilic, though maybe more clumped or ropey than perfectly glassy sometimes. But the composition is the key difference. It's made up mostly of tangled up bits of the cell's own internal scaffolding, specifically keratin intermediate filaments.
Whoa, okay. So this isn't protein coming in or going out. This is the actual structural framework of the cell itself that's become damaged and clumped together, the cytoskeleton.
Exactly. The internal architecture of the hepatocyte is breaking down. These keratin filaments normally provide strength and shape to the cell. But chronic alcohol exposure, likely through things like oxidative stress and metabolic disruption, causes these filaments to become chemically altered, misfolded, and aggregated.
So this type of hyaline accumulation signals a much deeper problem for the cell. Not just overload, but actual structural damage.
Absolutely. It signifies a profound internal crisis. The cell's structure is failing. This is generally a marker of more severe and often irreversible injury compared to, say, the reabsorption droplets.
And you mentioned earlier this connects to a broader idea about cytoskeletal proteins accumulating.
Yes, it does. It helps us see that hyaline change isn't just about soluble proteins. It can happen whenever the cell's structural components get messed up. The classic parallel here is a neurodegenerative disease. Think about the neurofibrillary tangles seen in Alzheimer's disease.
Right. Those are made of tau protein, aren't they?
Exactly. Tau protein is associated with microtubules, another key part of the neuron's cytoskeleton. When tau gets abnormally modified, hyperphosphorylated, it clumps together, forming those tangles. And guess what? Those tangles are also eosinophilic, appearing as intracellular inclusions.
So whether it's keratin filaments in the liver due to alcohol or tau protein in the brain in Alzheimer's, the end result, this eosinophilic hyaline-like clump, signifies a breakdown of the cell's internal support system.
Precisely. A much deeper and usually more permanent kind of cellular pathology than just having too much protein traffic.
All right, let's step outside the cell now. Extracellular hyaline. You suggested this often points towards more chronic, maybe even permanent changes, things that have been happening over a longer time.
Generally, yes. When you see this glassy pink material accumulating in the extracellular space between cells, it often tells a story of chronic injury, tissue remodeling, or long-standing systemic problems affecting tissues.
Okay, what are the key examples here?
Well, there are two main ones we need to cover. The first is maybe less clinically dramatic, but interesting conceptually, the hyalinization of scars.
Hyalinization of scars. So old scar tissue can start to look glassy and pink.
That's the observation. In very old, mature scars, the dense collagen fibers that make up the scar tissue can sometimes lose their distinct fibrillar appearance and take on this more homogeneous, smooth, glassy, eosinophilic look.
Seems straightforward enough, just collagen aging or something.
That's where it gets a bit fuzzy, actually. The source material we look at makes a point of highlighting this. While pathologists see this change, the exact biochemical basis for why old collagen fibers sometimes look hyalinized isn't fully understood.
Oh, really? So we don't know exactly what's happening chemically?
Not precisely. There are hypotheses, maybe about increased cross-linking between the collagen molecules over time, or some kind of denaturation that makes them pack differently and stain more uniformly. But the bottom line is, calling an old scar hyalinized is still mostly a visual description in this context, because we haven't nailed down a single definitive molecular change that causes it.
Interesting. So even outside the cell, the term can be purely descriptive sometimes.
Yes. But that brings us to the second, and arguably much more clinically significant type of extracellular hyaline, arteriolar hyalinization.
Arteriolar hyalinization. Okay, arterioles are the very small arteries, right? The resistance vessels.
Exactly. And this type of hyalinization, the deposition of glassy pink material, right in the walls of these tiny arteries is a huge deal clinically. It's particularly common and damaging in the kidneys.
And you see it in specific diseases.
Very much so. It's a hallmark finding in patients with long-standing hypertension, high blood pressure, and also in diabetes mellitus. These are two massive public health problems, and this microscopic finding is central to how they damage organs like the kidney.
Okay, let's break down the mechanism here. How does this glassy pink stuff actually get into the wall of the arteriole? It's not inside a cell anymore.
Right, it's happening within the layers of the vessel wall itself. And it seems to be driven by two main things happening together, especially under the stress of high blood pressure and diabetes.
Okay, what's the first?
The first is basically leakage from the blood. In chronic hypertension, the constant high pressure pounding against the inner lining of the arteriole, the endothelium, damages it. This makes the vessel wall leaky.
Leaky to what?
Leaky to proteins circulating in the blood plasma. Things like albumin and other plasma proteins get forced out of the bloodstream through the damaged lining and seep into the actual wall of the arteriole. The layers called the tunica intima and tunica media. We call this extravasated plasma protein.
So plasma proteins are literally oozing into the vessel wall because of the high pressure. That's part one. What's part two?
Part two involves the cells within the vessel wall, particularly smooth muscle cells, and it's really prominent in diabetes. Chronic high blood sugar, hyperglycemia, and all the metabolic changes that come with it seem to stimulate these smooth muscle cells.
Stimulate them to do what?
To produce excessive amounts of extracellular matrix components, including basement membrane material. Basement membrane is normally a thin layer supporting the cells, but here they start laying down way too much of it.
Ah, okay, so you've got plasma proteins leaking in from the blood and the vessel wall cells producing extra structural material.
Exactly. And both of these components, the leaked plasma proteins and the excess basement membrane material, are protein-rich and therefore stain pink with eosin. When they accumulate together in the vessel wall, they create this characteristic thick, homogeneous, glassy pink appearance. That's arteriolar hyalinization, or sometimes called hyaline arteriolar sclerosis.
Thick, glassy, pink walls in the small arteries. Hey, now connect that to the clinical impact. Why is this thickening so bad, especially for the kidney? This is the so what?
This is absolutely the so what.
Yeah.
Think about what an arteriole does. It controls blood flow into the tiny capillary networks downstream. The kidney is basically a massive collection of these tiny capillary filters, the glomeruli.
Right.
If the arteriole bringing blood to the glomerulus has its wall thickened by this hyaline material, what happens to the channel inside, the lumen, where the blood flows?
It must get narrower.
Dramatically narrower. This hyaline deposit significantly narrows the lumen. And if the pipe gets narrower, less blood can flow through it.
So less blood flow to the kidney's filtering units.
Exactly. This leads to chronic ischemia, a persistent lack of adequate oxygen and nutrients for the parts of the kidney supplied by that narrowed arteriole. Over months, over years, this chronic starvation causes the downstream kidney tissue, the tubules and glomeruli, to wither and die.
And that leads to kidney failure eventually.
It's a major pathway to it. Microscopically, this chronic ischemia causes the renal tubules to shrink and disappear. That's called tubular atrophy. And the spaces where they used to be fill up with scar tissue interstitial fibrosis. The glomeruli themselves can also scar down glomerulosclerosis. The cumulative effect of all this scarring, driven by the narrowed arterioles, is called nephrosclerosis.
Wow, so that little patch of pink, glassy material in the wall of a tiny artery is basically a visible sign of years of damage from high blood pressure or diabetes, directly leading to the gradual destruction of kidney tissue.
It captures the whole story. The hyaline change itself diagnoses the process hyalinization. Its location in the arterial wall points strongly to the systemic causes hypertension, diabetes, and it directly explains the long-term consequence, reduced blood flow, leading to nephrosclerosis and kidney damage. It's incredibly informative.
Okay, so if we pull back a bit and try to synthesize all this, hyaline change, whether inside or outside the cell, seems to sit right at the intersection of abnormal accumulations, especially involving proteins.
That's a great way to put it. It's a key morphological pattern that arises from various ways the body mishandles proteins or structural components. And, advances in pathology aren't just about describing these things anymore. They're about understanding the underlying molecular failures.
The big lesson seems to be, see hyaline, think protein problems. Either too much normal protein, problems moving it around, or maybe fundamental issues with the protein's shape or structure itself.
That really is the core message. Modern biology helps us categorize these failures. You can have accumulation because normal stuff isn't being removed properly, like maybe lysosomal storage diseases, although that's a different topic. Or you can have defects in protein folding and transport, which is highly relevant here. Or failure to degrade abnormal metabolites.
And you mentioned misfolded proteins being a key area of advancement.
Yes, understanding what happens when proteins don't fold correctly is crucial. Let's use a classic genetic example that really illustrates this. Alpha-1 antitrypsin deficiency.
Ah, yes. I remember this involves both the liver and the lungs, right?
Exactly. Alpha-1 antitrypsin, or AAT, is a protein mainly made in the liver. Its job is to travel to the lungs and protect them from enzymes that can damage lung tissue, like neutrophil elastase.
Okay, a protective protein made in the liver.
Right. But in the most common form of AAT deficiency, there's a mutation, often called the Z allele. This mutation screws up the protein structure.
So the liver cell makes the AAT protein, but because of the mutation, it can't fold into the correct three-dimensional shape inside the endoplasmic reticulum.
Precisely. It gets stuck in a partially folded, misbehaving state. And worse, these misfolded AAT molecules tend to clump together, they polymerize, forming aggregates inside the ER of the liver cell.
So they can't move on to the Golgi, they can't get secreted out of the liver cell to go do their job in the lung?
Nope. They're trapped.
Hmm.
And this mass of aggregated, unsecreted protein builds up within the hepatocyte cytoplasm. And guess what those inclusions look like under the microscope?
Let me guess. Pink and glassy.
Yeah,
Hyaline-like.
bingo. They stain strongly eosinophilic and are often described as hyaline-like globules. So the disease has two arms. The lungs suffer because they lack the protective AAT enzyme, which leads to emphysema.
You're right.
But the liver cell suffers because it's choked with its accumulated, aggregated junk protein.
That really highlights a modern concept in pathology, doesn't it? Disease isn't just caused by losing a protein's function somewhere else. The cell making the bad protein can also get sick from the buildup.
That is a profoundly important insight. It's called ER stress. The endoplasmic reticulum, the protein folding factory, gets overwhelmed by all this misfolded garbage.
ER stress, like the cell's internal quality control system is sounding the alarm.
That's exactly what it is. The cell senses this logjam of unfolded protein in the ER and triggers a defense mechanism called the unfolded protein response, or UPR.
What does a UPR do?
Initially, it tries to fix things. It might boost the production of helper proteins called chaperones that assist folding. It might also temporarily slow down overall protein production to reduce the burden. It's trying to restore balance or protein homeostasis.
But what if the problem is too severe, like with this AAT mutation? What if the UPR can't clear the jam?
If the stress persists and the cell just can't cope, the UPR shifts gears. It concludes the situation is hopeless, the damage is too great. And then, this persistent ER stress actually triggers the cell's self-destruct program. Apoptosis. Programmed cell death.
Wow. So in the AAT deficiency case, those glassy pink globules aren't just inert stored stuff. They're a visual sign of a chronic, toxic internal crisis that is literally pushing the liver cell towards killing itself. That's a world away from those reversible kidney droplets.
That distinction is absolutely critical for anyone learning pathology. When you see hyaline change, you have to ask, what kind is this? Is it potentially reversible, like an overload of normal protein that can be cleared if the stimulus stops, like the renal droplets? Or is it an accumulation of abnormal misfolded proteins, like AAT, or damaged cytoskeletal elements, like alcoholic hyaline, or irreversible deposits in vessel walls, like arteriolar hyalinization? These latter types signify severe cellular stress, structural damage, or permanent systemic changes. That difference often determines the prognosis.
It really elevates the term hyaline, doesn't it? From just a passive description to an active clue about underlying molecular chaos and systemic disease. Hashtag ouchag outro.
Okay, that was a fantastic deep dive. We really unpacked a single term, but found so much clinical and molecular meaning behind it. Let's quickly lock in the main takeaways.
Absolutely. Takeaway number 1. Hyaline means a homogeneous, glassy, pink appearance under the microscope with H&E stain.
But crucially,
It's descriptive, not diagnostic on its own. It signals something is accumulating, often protein-related, but you need more info.
right, and we saw it mainly in two locations. Inside the cell intracellular. Examples there were the reversible reabsorption droplets in the kidney due to protein overload.
And Russell bodies and plasma cells from synthesizing too much normal antibody protein.
Then we contrasted those with alcoholic hyaline, Mallory bodies in the liver, which is different. It's tangled cytoskeletal filaments signaling deeper cell injury.
Exactly. And then outside the cell, extracellular, we touched on hyalinized scars where the mechanism isn't fully clear. But the really big one clinically was arteriolar hyalinization, that thickening of small artery walls, especially in the kidney.
Driven by protein leakage from hypertension and excess matrix deposition in diabetes. And the major consequence is narrowing of the vessel lumen.
Leading to reduced blood flow, chronic ischemia, and ultimately nephrosclerosis and kidney damage.
You got it.
Fantastic. Thanks for walking us through all that microscopic detail and connecting it so clearly to the bigger picture. Before we sign off, what's one final thought for our listeners to chew on about hyaline change?
I think it's worth reflecting on the permanence of some of these changes, particularly the extracellular ones like in the arterioles. That hyaline material accumulating in the vessel wall is basically there for good. It's not getting metabolized away like those kidney droplets. So that tiny patch of glassy pink stuff becomes a permanent physical scar tissue, a microscopic record etched into the body, documenting years of maybe uncontrolled high blood pressure or high blood sugar. It really forces you to see how chronic systemic conditions leave lasting structural marks. And those marks are ultimately what drive organ damage and disease progression over time.
A microscopic history book written by chronic disease. That's a powerful image. Really fascinating stuff. Thank you, everyone, for joining us for this deep dive. We hope it sheds some light on this fundamental concept. We'll be back soon with another exploration.